Sure, we have a bunch of different promising new chemical and new molecular entities that are coming onto the scene that may be effective for acute or chronic pain. And what we're finding is some of these do not have the traditional mu opioid receptor type of activity, both from a pharmacodynamic effect of efficacy and side effects. And some of them may not have the potential societal issues that we would see with traditional mu pure opioid agonist. An example of this is the kappa agonist with Cara Therapeutics. This is a very interesting compound that is moving through its developmental phases now, and this compound may provide analgesia through a peripheral action on the kappa receptors, and this may mitigate or decrease the central nervous system effects, which could be very promising. So that's one that we're looking at. There's another one that looks at ligand type based science, and this company is based in Philadelphia, and they have, again, another potential new chemical entity or new molecular entity that is going to work on a different mechanism of action, still utilizing that type of pain pathway that we would with other types of analgesics, but in a different way, again, with potential mitigation of side effects, particularly those that may lead to the unwanted behaviors that we see with some of the pure mu opioid agonists. We have another molecule that you can say has been out there for a while, but is the only real new product in its class of opioid agonist, and that is tapentadol. It's been around since 1995, and this molecule we're learning more and more about. And I think what we're seeing is that the norepinephrine reuptake inhibition of this molecule, coupled with the mu opioid agonism, might allow us to address different types of pain that have a multifactorial etiology. So those that may have symptomatology of nociceptive and neuropathic type pain. There's another drug too that's coming out that is trying to focus as a fixed dose combination of an opioid and an antiemetic. If we look at opioid-induced nausea and vomiting, what we find is that this is one of the most common side effects, particularly with acute exposure to an opioid. So some potential way to mitigate the effects of the opioids in the enteric nervous system and in the central nervous system that result in nausea and vomiting might also be promising. So that might be coming to the market soon as well. If we look at trial design, what's happened is if we are going to think about developing a drug class that is considered to be opioid, then we're going to have to look at potential human abuse liability trials. And the FDA now does have some finalized guidance for branded opioids on human abuse liability, or what we call abuse deterrent formulations. We also have some draft guidance on the generic type of abuse deterrent formulations. And so what we're seeing is a better understanding of what Section 9.2 in the full prescribing information or package insert is, that describes what type of testing has been done to give us an understanding of whether or not these agents and based on their physiochemical activities might lead to less ability to extract the active product ingredient, and then through the clinical trials find out the likability or take drug again. So I think probably the biggest thing for this class of drugs is that we do now have finalized abuse deterrent guidelines for the opioids, and we have a draft guidance for the generics. A lot of people ask when they look at drug development, particularly when we use an older drug and clinically repurpose it. This pathway is normally called the 505(b)(2) pathway. Now, sometimes it may not fall into that, but this 505(b)(2) pathway means that we already know a lot about the drug. It's been around for a long time, but now we're using it a different way. We're using it for pain relief, or we're administering it a different way. And the burden of proving new information really becomes focused on certain elements, like what are the blood levels, so the pharmacokinetics of the drug. Or if it's for a certain indication like back pain, how does it work in back pain if it hasn't been tested formally before? We have different types of trial designs we can use. Right now, it's very common that we use an enriched enrollment randomized withdrawal trial, an ERW type trial. And what this allows us to do is a couple things. One, it allows us to understand what would happen in a situation when we titrate a patient to effect. And then when we decide if the patient has shown that the drug works and they can tolerate it, we'll switch them to placebo, or we'll randomize them to a placebo arm, or we'll keep them on that steady dose, and we'll look at those patients who then would withdraw, either for lack of efficacy or adverse events. So this gives us a good understanding of, one, the titratability of the molecule, particularly in opioid-naive patients, and number two, the durable analgesic effect. So it's a very commonly used development type of trial. It may not be what we would do in a phase 4 trial, where in a phase 4 trial, we may be saying, it's much more important to understand functionality, activities of daily living. So those are our primary endpoints. And pain scales, pain, just because you reduce pain doesn't mean you have improvement, what some people might say. So it's different than a trial that we might look at for outcomes that clinicians may feel might be more directional for them. So this is, the dichotomy that exists right now when it comes to developing drugs, is that we basically have a set of blueprints, in a sense, that you can follow, and that might come out with results that show there's a reduction in mean baseline intensity, for example, but might not be transparent when it comes to the effect on improving functionality, quality of life, or activity of daily living. So it's not uncommon that the clinician comes to me and says, Joe, when you've got your drug development hat on, why don't you do this and why don't you do that for the study? Part of it is, remember that these are not purely scientific in nature. There is a commercial element behind drug development. And like anything else, you want to risk mitigate that process so that you will get approval. And then hopefully the more responsible companies, when the drug does get approved, they do allow for principal investigator-initiated trials, like those of my co-chair, Dr. Shireen Alamachu. He does quite a few of those studies that try to answer the question that the everyday practitioner would like to know. Sure. So when we look at new targets, the question is, are we looking at a receptor? Are we looking at an enzyme? I'll give you one example of a target that I like. We think about opioids, and everyone talks about opioids and potential problems with them. But when you really get down to it, the opioid, the class of receptors in our body, these receptors are really, through enkephalins and endorphins, are the natural way our body takes care of pain. So it's a very natural process. We have synthetic opioids, yes, but the body is engineered to utilize those receptors in different ways, and one of the ways the mu receptors are used, as an example, are for analgesia. So if there were a way to use our own body's natural opioids, keep them around longer, make them stronger, then maybe we do not need to use synthetic ones or introduction of non-produced opioids in your own body. So the DENKIs, the dual enkephalin inhibitors, are very interesting. They go in and they try to, in a sense, knock out the enzyme that would break down your opioid, your own endogenous opioid. And this could be really exciting if we could accomplish that, if they do help. Yeah, and it would be able to use your own opioids to address the pain. Right now, those opioids usually are not strong enough. You know, when you hurt yourself, they get expressed, right? But they usually, for very severe pain, they may not be enough, and they may not last long enough. So these are some of the targets that we might look at. There's an example of it.